So this is a GCSE session on microscopes. There are 5 questions all relating to cells. Have a go at the 5 questions. Pause the video and you're ready, you can restart and check your answers. So pause now. So question number 1, name the part of the cell linked to aerobic respiration is a mitochondria. Remember GCSE, you need to know aerobic respiration is mitochondria, but anaerobics in the cytoplasm, which is why cytoplasm is common to all cells. What animal cell is adapted to have no nucleus? It's the red blood cell. Name of the small circles of DNA in a bacteria cell are called plasmids. 3 structures found in all cells. All cells have a cell membrane, cytoplasm and ribosomes. And the name of the 3 structures found in plant cells but not animal cells are the cell wall, the chloroplast and the permanent vacuole. So one of the skills you'll need for your GCSEs is to turn numbers into and out of standard form. So some of you may already know how to do this, in which case you can just do the questions. For those of you who don't, I'm going to show you a quick and easy way to turn numbers into standard form and out of standard form. So in order to do standard form, you need to be able to get numbers and turn them into standard form. So you write the number out and you write the decimal place in the number as it starts off. So for the number 785623, the decimal place is after the 3. Then what you do is you put the decimal place in a new position where you will end up with a number between 1 and 10. So in this case, the new number would be 7.85623. You then have to work out how many steps you've moved the decimal place from its original point to where it gives you the number between 1 and 10. In this case, we have to move a total of 5 steps. And we then put that down as 10 to the power of 5. Now, if our original number is a decimal number, we do all the same steps. We write our number out, we put where the decimal place was in the original number, we put where it goes to get a number between 1 and 10, we count the number of steps we have to move, but when we write out our standard form, instead of 10 to the power of a whole number, we do 10 to the power of a negative number. But it's exactly the same process up to that last step. If we are given a number in standard form and we want to turn it into its original number, we simply do the reverse process. So 6.957, we have 10 to the power of 8. That means that we're going to move the decimal place 8 steps. And because it's a positive number, we're going to have an original number which is really, really big. It's not going to be a decimal. Whereas if you look at the other example, where 5.4687 by 10 to the minus 4, because it's a minus number on the power of 10, we know the original number is going to be a decimal point. So we write out our number, 6957, with the decimal place in its original position. We add loads of zeros, so we can add 8 zeros onto the end because it's 10 to the power of 8, we just add in 8 zeros. But then we have to move the decimal plate 8 steps to make sure it ends up in the right position. So it doesn't matter how many zeros we add onto it on the right, as long as we only move 8 steps, the decimal place will end up in the right place, and then we can write out the number. In the negative number, we add the zeros, but we add it to the left of the original number because our new number is going to be a decimal. We need all the zeros at the start. Whereas when our new number is going to be a large number above 0, we need to put the zeros on the right hand side. So now you've got a couple of examples where you have to turn numbers into standard form and you have to turn numbers out of standard form. Again, it doesn't matter what exam you're doing, it's more than likely you'll be expected to turn a number in one of your science exams into or out of standard form, more likely into. So have a go at the questions. If you are getting them right, move on. But if you are struggling and you haven't watched the earlier bit of the video on how to turn numbers into and out of standard form, I suggest you go back and make sure you can do this skill. So here are all the answers. Note the negative values where we're involved with decimal numbers, and obviously in the second part where you had to turn numbers out of standard form, you end up that if you had a minus number in the standard form, you have a decimal number in your final answer. So another skill you're going to need to get through your science exam is the use of prefixes on certain units. So prefixes are words we put in front of other words to give us a different version of the unit. So there are a large number of these, but the main ones you're going to need for your GCSEs are the words centi, milli, micro, and nano. And you can see we've got each of the symbols for each of those words. When you're converting from units, I would always suggest that you write down one of the larger prefix, for example, 1 centi, and then you need to have memorized how many millis are there in a centi. So 1 centi equals 10 millis. So when it says larger prefix in the bottom right hand corner, that would be the number 1 centi, and the smaller prefix would be 10 milli. And then you simply have to work out if you are timesing by 10 or dividing by 10 to get from the number 1 to the number 10. So if we're going from centi to milli, we are timesing. If we're going from 10 milli back to 1 centi, then we have to divide by 10. And it's the same pattern for all of them in terms of moving backwards and forwards to work out if you're timesing or dividing. 1 milli is 1,000 micro, so you've got a times to go from milli to micro, and 1,000 micro is 1 milli, so you have to divide to go from 1,000 micro to 1 milli. So now we've got a couple of examples for you to practice on. So pause the video, have a go at the questions, and whenever you're ready you can restart and check your answers. So pause now. So first off, to convert the 20 millimeters into the nanometers, well, we are going from a larger prefix to a smaller prefix. So in this case, we're going to be timesing by a certain number. And rather than try and do it all in one go, we can simply go from millimeters down to micrometers. We've got to times by 1,000, and then to go from the micros to the nanos by timesing by 1,000 again, which gives us a value of 20 million. When we're converting millimeters into micrometers, again, we're going from a larger prefix to a smaller prefix. So we're going to times, but this time we are just timesing by 1,000 because we're only going one step. That gives us an answer of 15,000. When we're going from a smaller prefix, so one on the right hand side of the list, to a larger prefix, one on the left hand side of the list, then we are going to be dividing. So the 10 millimeters into centimeters, we simply divide by 10, which gives us an answer of 1. To get from nanometers to millimeters, you have to take a couple of steps. So you divide by 1,000 to get you to the micro, and then divide by 1,000 again to get you all the way to the milli, gives you an answer of 0.0001. And then in the last question, to get from the 25 micro all the way up to the centi, you've got to divide by 1,000, and then we divide by 10 to turn the milli into centi. That gives you an answer of 0.0025. So compound microscopes date all the way back to the 1590s, and they have improved in terms of the technology and the quality of lenses and electricity and lighting and so forth, but for the most part the basic elements have stayed consistent all the way through. We've just been able to improve the quality of the resolution so we can see things in much more detail. So one of the things you're going to have to do is you're going to have to memorize the parts of a microscope. Now some of these are more likely to form part of an exam question than others, and I'll go into that in a little more detail in a second. But for now, what you want to do is you want to write down the numbers 1 all the way up to 9 and use the words on the right hand side and try and match them up to your answers. So pause the video, have a go, see how many you can do. If you're really struggling, continue with the video, get the answers, memorize them, and then go back again, replay the video and see if you can do it from memory. So pause the video now. Well, the answers are as follows. And there's only certain ones that are going to appear on the exam. So the most obvious ones to appear on the exam are things talking about the focus, things talking about the objective lens, things talking about the mirror and condenser, which controls brightness of light, and possibly the stage clips to hold the actual slide in place. But they're the most likely questions you're going to be asked about. So what are they actually going to ask you? Well, In an exam, you may be given a picture or you may be given some details and you may be asked, how would you make this image better? Well, if the image is blurry, if it is out of focus, so what we would do is we would use a focus to fix it, but we need to be specific in which one. Most of the time, if an image is very badly blurred, we would start with a coarse focus, and we always wind the stage so it's closest to the lens, the objective lens, to make sure we don't break any slides or scratch any glass. But we'd start with a coarse focus. The fine focus, really we would only use when things are just about perfect. We're just trying to get that little bit of detail on it. Most of our focusing will be done with a coarse focus. If, however, we want to magnify the image, we want to see it essentially bigger, then we need to swap over our objective lens because that's what we use to magnify it. And then, as I said previously, anything in terms of lighting, you're going to come down to the mirror to turn light on and off, or electric lamp if it's a bulb underneath. And a condenser will actually let us turn the brightness up and down. So all microscopes can be broken down into two main categories. We have our light microscopes, and our light microscopes are really useful for seeing things that are alive, and they're really useful for seeing things in color. But they don't give us a huge amount of detail. So what we can use is we can use electron microscopes. And electron microscopes give us what's called a much higher resolution and a much higher magnification. And there are two types of electron microscopes. There's one called a TEM, which is where the electrons go through the sample in the same way that the light rays go through a sample in a light microscope. But what we also have is a SEM, which is a scanning electron microscope rather than a transmission electron microscope. And the scanning electron microscope just scans the surface of anything that's put in as a sample. So there are two words that I've just used. They're magnification and resolution. If I make a picture bigger in size, I have increased its magnification. But I can make pictures bigger and I can start making them very pixelated because what I haven't done is I haven't increased the resolution. Resolution allows us to see more detail in anything, whereas magnification simply makes it bigger. And most of the time when it comes to microscopes, it's an increase in resolution that gives us more detail about what we're actually seeing in the sample. Now, in terms of improving magnification and improving resolution, both cases we have the same practical answer. We simply use an objective lens with a much higher power. So on an objective lens, normally in a light microscope, you will have at least 3 lenses, but you can have other versions. You can also take lenses off and replace them with more expensive ones. But the statement you have to use is higher powered objective lens. You have to tell them that it's higher power, and you also have to tell them which lens, because remember there is also the eyepiece lens in there, so you got to be specific and say objective lens. So it's not impossible you'd be asked to either draw some sort of microscope drawing in an exam, you may be asked to label something, or you may be asked what's wrong with a particular drawing that somebody else has produced. Things to look for are shading and coloring is a no. There should be no shading and no coloring. We use single thin pencil lines, that's it. There's no multiple brush strokes or any kind of fancy drawing lines. It's just pencil on paper and draw single lines. We also have to make sure when we're doing our labels that we should use a ruler to draw a straight line, preferably horizontal, but they will not mark you down in an exam if it wasn't a diagonal line or any kind of straight line in any direction, but they really should be horizontal. And they definitely do not have arrowheads. When you're drawing a label line, you go from the words that you've written and you draw directly, and the line must touch whatever it is that you are trying to label. So label lines must touch. They don't need arrowheads or anything on the end of them. And in terms of your drawing, you want a very clear drawing, no shading and no coloring. One of the most common questions in the exam is about the magnification of a drawing or the magnification of something on a slide and how you move between those two numbers. And it's using what's called the I-am equation. It's got the letter I on the top of what I refer to as a Smurf, simple method for understanding and rearranging formula. You may see a similar type diagram when you do your math lessons, which is a triangle. The difference is the addition of the two division and the times symbols, just to make it easier for you to work out what you do with the numbers. And it's always the top row divided by the bottom row, or the bottom row times the bottom row. So in this case, it would be the image size divided by the actual size, or the equation would be image size divided by magnification, or it would be the actual size of the thing on the slide multiplied by the magnification. In this example, it says the actual size of the nucleus in the diagram is 6 mu m. Remember, the mu is the micro. What that means is the black line going through the center of the nucleus, which is the gray patch in the bottom right hand corner, that black line is representing the distance of the nucleus. If you see something like that, they will normally ask you to get a ruler and to measure that distance. Of course, the issue being that your ruler will measure in millimeters. So you have to get your millimeters, whatever number of millimeters the drawing is, and convert it into mu m. If you've forgotten how to do that, go back to the earlier part of the video. When you've done that, you should be able to calculate the magnification by getting the size of the image, which is the picture you've just measured in millimeters, and then convert that into mu m by times it by 1,000. And then what you can also do is take the 6 mu m, which is the actual size of the slide, and it's the number you got from the picture on the paper divided by the actual size, which will give you the magnification. And it will very much depend on how big your screen is, because as we said, we can make the screen bigger and smaller, that's magnification. But we may not see more detail because that's resolution. So here's a couple of questions, have a go at. Some of them do involve converting units as well, so make sure you know what you're doing converting the units before you attempt any of these questions. So let's work our way through the answers. So on the first one, we have a cell nucleus that measures 30 mm, which is millimeters, and it have an actual size of the nucleus is 6 mu m, which is our micrometers. So because we got millimeters and micrometers, we need to convert them into the same unit. So the 30 millimeters, to turn them into the micrometers, we have to go from a big unit to a smaller unit, so we are timesing. And one step we are timesing by 1,000. So the 30 becomes 30,000. Then what we need to do is we use our IAM equation. The 30,000 is the actual image size, so that's it on the diagram. So that's I, and it gets divided by the actual size of the nucleus on the slide, which is the 6 mu m, which gives us an answer of 5,000. So our magnification is 5,000. We could also turn the 6 mu m into mm and do the same division with the image divided by the actual, but you will get the same answer. So in this question, again, we're talking about chloroplasts this time, and it doesn't matter which part of the cell or even the whole cell we're talking about, it's always the same sequence of steps. So we've got an image size, a drawing of 25 mm, which is our millimeters, and we've got a magnification of 2,500. Well, this time we don't have to worry about things being in the same unit, because one's a magnification and one is the image size. So we can safely 25 millimeters, which is the size of the image, divided by the 2,500, which is the magnification, gives us an answer of 0.9 millimeters. However, we may have to convert that into different units at some other point, so it's always worth checking the question when you're finished off doing the calculation. Check to see if you need to give anything in different units or if you need to do anything in standard form. So in this question, we have an image which is 15 millimeters long, and we have the actual size is 5 mu m, so we need to get those both into the same units. And it doesn't matter if we use mm's or mu m's, as long as they're in the same unit, that's all that matters. So we'll convert the 15 millimeters into mu m, because it's just easier to times by 1,000 than it is to divide, and we get 15,000 mu m. And then we do the size of the image divided by the actual size of the thing on the slide, which gives us a magnification of 3,000, hence times 3,000 is your answer. So in this particular question, a image has been measured as 6,000 μm. Don't always assume that the image will be measured given measurements of millimeters. It can be given in any of those measurements and you have to convert them. Whenever we get the actual size of the cell, it says it is 2 millimeters. And again, it does not matter which unit we convert them, as long as both numbers are in the same unit, that's all that matters. But if we convert the 2 millimeters into μms, we do 2 times 1,000. That gives us 2,000 μm. Then we then go to our triangle and the image is on the top row divided by the actual size, which is on the bottom row to calculate the magnification, which means it is 6,000 divided by 2,000, which gives us a magnification of 3. So in question 5, we have an image of again another cell. At this time it's 4 centimeters in the drawing. So the image size is 4 centimeters and the actual size is 20 μm. So what we have to do is get them into the same units, just like before. Well, we convert the centimeters into millimeters by times and by 10. And then we convert from millimeters into μm by times and by 1,000. We can do that as 2 separate steps, which will give us a value of 40,000 μm. So our 4 centimeters converts into 40,000 μm. We then have our image size of 40,000 μm divided by the actual size of the object, which is 20, which gives us a magnification of 2,000, which is why our answer is times 2,000. Now there's a lot of maths in this, so please do go back through all the different things you have to do in terms of you might have to use numbers in standard form, you might have to convert units from one prefix into another, and obviously the I am equation is a very popular exam question as well. Best of luck.